The Neuro Growth Factor (NGF) by Chris Fry, Sally Yu, Randall Harley, and Jerry Shong.
History of NGF. In the 1940s, Victor Hamburgers invited Rita Levi-Montalcini and Stanley Cohen to work in his lab in St. Louis, Missouri at Washington University. Rita Levi-Montalcini studied abnormal growth of the ganglia, which is nerve tissue, in chicks due to tumors. At the time, she did not know if the tumors directly stimulated nerve growth or if they simply disrupted the normal development. She discovered that the tumors actually directly stimulated nerve growth. Then she isolated both the tumors and the nerve cells and only connected them by a diffusible membrane, and she still observed nerve growth, thus proving that the tumors do not have to directly contact the nerve cells, and determined that there must be a diffusible molecule, which they called neuron growth factor, NGF. The next step was isolating NGF, which was done by Stanley Cohen. He determined NGF was composed of mostly protein and some nucleic acid. Then he added snake venom to the purified NGF to degrade the nucleic acid and tested whether the NGF still worked. The NGF still stimulated nerve growth, proving that it must be a protein opposed to a nucleoprotein.
The structure of NGF. The structure of NGF was first found by X-ray and published in 1991 by MacDonald. As you can see in the picture on the right, it is a protein structure mainly made out of beta-pleated sheets. At one end on the left of the picture, cysteine knot motifs are formed, which are structures linked by this disulfide bond. The other end on the right, there are hairpin loops structures. NGF's primary structure includes 118 amino acids, and as I mentioned before, the secondary structure is mainly beta-pleated sheets. And the tertiary structure was formed by disulfide bonds that link the sheets together. Most NGFs are dimeric, which means protein structure formed by two subunits. From the publication of UCSD and other research institutions, we know that NGFs function with at least two classes of membrane receptors, the P75 neurotrophin receptor and TRKA, a transmembrane tyrosine kinases, both of which are associated with neurodegenerative disorders.
And now for the function of NGF in the body. NGF supports brain cell survival and growth of cell connections during fetal development. Until as recently as 10 years ago, researchers believed NGF's effects to be solely within this growth period. However, new research has produced evidence that NGF has effects in the adult nervous system as well. It boosts nerve cell activity and function in the fully functioning adult nervous system, both central and periphery. Specifically, it boosts axonal growth via increasing elongation and branching. Write that down. NGF also aids in the healing process of damaged neurons. There is evidence that NGF circulates throughout the entire human body and is important for maintaining homeostasis.
There are generally three phases of medicine clinical trial. No, you don't have to write this down. Phase 1 is to determine the stability and safety of a potential medicine in animal bodies. Phase 2 to supply the medicine to human body to see whether it's effective. In the last phase, phase 3, the medicine is applied to a larger group of patients. NGF is currently at the phase 2 trial. A phase 1 study of NGF in Rush University and the University of California, San Diego indicated that a single administration of the therapy was generally safe and well tolerated. A phase 2 multicenter trial of NGF gene therapy is scheduled to begin later in 2008. The trial will include a placebo group and will be performed in a larger number of people, allowing a more clear understanding regarding the potential ability of NGF to treat Alzheimer's disease.
So, in Alzheimer's disease, deep brain cells called cholinergic neurons are lost, and these cells are NGF responsive. The loss of these cells contributes to cognitive decline in Alzheimer's disease. Knowing that the cholinergic cells respond to the neural growth factor, scientists modeled the neuron loss in monkey brains by cutting their connections to the hippocampus. Each black dot in the picture represents one cholinergic neuron, 1 month later, noted by the picture, they noticed a significant loss of the neurons. However, the reintroduction of a neuron growth factor after the damage demonstrated the protection of the neurons. In the graph, without neural growth factor, 75 of the neurons died, and with the growth factor, 80% of them survived. The neural growth factor prevents death of neurons in brain as it reverses the atrophy of neurons in aging. It actually prevents cell degeneration.